Hi there, Mr. Bleeker again. We'll get going on our next lesson in eCampus Live Biology. We'll start in on the nutrients. We'll begin with carbohydrates, and then move on to lipids and proteins, and that pretty well finishes off our biochemistry unit. We have a lot of ground to cover, for example, how these molecules synthesize or come together, and how they're torn apart. I'm just going to predicate all this with a little piece of information, and that is it involves the addition or removal of water, specifically by enzymes. It's an interesting trick, so let's get there right now. As a quick aside, if you have a chance, the YouTube channel is live, and a lot of students just like to go directly to the YouTube channel. You can do that by creating a bookmark or going through the course site. It's up to you. Neatly arranging them into lessons 1, 2, 3 as we go through the course, so trying to make it a little more convenient for you. Okay, so let's get to it. And we are there now. Excellent. Okay, there is a major laboratory component for this section where we'll identify all the different sort of biological molecules, carbohydrate test, lipid test, protein test, test for fats. I always love the test for fats. Wiping paper towel across your forehead will pretty well tell you whether or not something's a fat. Just look for the sheen that appears on brown paper. That's one of the easiest ones, so I'll give that away. But day one is carbohydrates, so here we go. Now, carbohydrate's an interesting term. It's sort of a little bit misgiving. People don't realize the name says exactly what it is. So if you think about the term carbohydrate, let's switch off red, not my favorite. So if we say, you hear carbs all the time. So I'll just there we go. Now, if you sort of glean in on two parts of this, you've got the carbo part, and then you've got hydrate. Now, that sounds kind of obvious, and it's supposed to be. That's really the derivative of the name, a carbon that is hydrated. It's a great way to remember it. So let's switch colors, and there we go. So if we take carbon, it's being finicky today. There we go. You take carbon and you hydrate it, this would probably be what you would expect. And that formula is not far off. What it should be telling you, you should be sort of discerning from it right now, is that it's 2 parts hydrogen to 1 parts carbon and oxygen. Now, you don't have to write this in, but I'm going to just for the sake of being obvious. There you go. A carbohydrate. Another way to write that formula is C to the N, where N is just however many carbons you have. You always have twice as much hydrogen, so there's that subscript, and you have the same amount of oxygen as nitrogen. So your formula for any carbohydrate is right there. So for example, if we looked at a 5 carbon carbohydrate, it would be C5H10O5. So nice that it's laid out that simply. It's very, very formulaic. Now, as far as molecules of life go, carbohydrates are largely energy compounds. And when we sort of look at carbohydrates, you can think of sugars being built up into starches, and breaking down starches to sugars is another way that our metabolism functions to give us energy, keep us alive. When you think about carbohydrates, I want you to picture someone shoving coal into a good old-fashioned boiler and burning the coal for energy and the release of carbon dioxide, and there's a side product of heat and a little bit of water is produced. See, that's not far off. That's exactly what happens in our mitochondria, which we studied in our cell biology unit. They're energy compounds. Feed these things to our mitochondria, and you get energy, or ATP, adenosine triphosphate, as a result. So there we go. General formula again for you. When we look at the different carbohydrates, We want to look at how many carbons they're composed of. For example, we'll just go down here. Glucose is a hexose sugar. So think of hexagons, for example, and that's not far off. When you look at the structure of glucose, and I'll just draw sort of a flat 2D version. There we go. You can picture glucose as being a hexose ring, like this. I won't draw on any of the side groups, but there you go. So sort of the structure of glucose. When we move into some of our other sugars, you're going to see what appear to be five-sided rings, but you have to pay attention to the number of carbons. It's not always the shape that determines whether it's a 6 or a 5 carbon unit. Now there's a little bit of terminology here. When we start studying carbohydrates, we have to talk about what they're made up of. So when we talk about the subunits of something, think of Lego, for example. Since carbohydrates are made up of sugars, we talk about monosaccharides. So monosaccharides would just be individual pieces of Lego, so single pieces. Mono meaning one, and saccharide meaning sugar. I remember when I was just a little guy going to my grandma's in Northport, Washington, and she always had this sweetener in her cupboard. It was called saccharin, and it was a sugar substitute. And I remember asking, Grandma, what does saccharin mean? And she said, oh, that just means sweet, dear. But really, when it comes right down to it, she was being sweet. But she wasn't far off, because it means sugar. So we have a couple of different units like this. Glucose and galactose both look like this, although some of their chemical groups are a little bit different. When you look at fructose, and we will, it appears to be five-sided. It's resisting me here. But then you'll look, I think I've got this position correctly, you'll see another carbon attached to the ring. So even though it looks five-sided, it's actually six-sided. A little bit of biochemistry here that you need to know about. Generally at the corners, when you look at these things, for example, generally at the corners, look carefully at the diagrams, and you'll see that at the corners, unless otherwise indicated, you'll see that the corners represent a carbon atom. It's sort of interesting here. It's being a pain, so I'm going to use my finger. There we go. So look at the structures, and then if there's a separate carbon atom to consider, in this case, you have to count them up. And you'll notice the way that I was drawing them, they actually go anti-clockwise. I'm not getting into the finer details here. That will come. But nevertheless, we'll get going. Okay? Let's pop along a little bit here. Just before I go on to the next slide, when you look at DNA, we studied a little bit of that in grade 11. For those of you just sort of remembering the structure of DNA, there's a phosphate group leading down to I'm not drawing it perfectly, but with my stylus I'll do my best here, a ribose or deoxyribose sugar, and then you go off to the genetic base, which could be adenine, guanine, cytosine, or thymine, depending on which base is in place. This sugar right here, this five-sided sugar, this is in fact a structural component of our DNA. It's interesting when you think that we use a 5-carbon monosaccharide to hold the edges of the DNA molecule together, but we do. And there's just two versions of that, whether it's oxygenated or deoxygenated. But for now, we'll just call it a ribose sugar. Whether or not we deprive it of oxygen, well, we'll talk about that when we get to RNA. Here's a nice up-close shot of it, for example. And it's interesting when you look at it. I'll count up it's a 5-sided sugar. There's carbon 1, 2, 3, 4, and as I say, you have to pay attention to if a carbon side group is going off there. It's a part of the molecule. So when you look at the carbons, you go in that direction. So that is at first glance, you'd look at the corners and say, hey, that's a a 4-carbon sugar. No, because you have to count the extra carbon going up and off the top. There we go. Now glucose is, now that it's drawn fully, I'll draw in the carbons. Glucose, let's just for the record identify this as the most important monosaccharide or single sugar that our bodies run off. If you are in the hospital and they put glucose IV in your arm, the reason they do that is because glucose is primarily what our cells run off of. Glucose itself can be broken down very efficiently into sort of smaller carbon chunks and fed into our mitochondria. Glucose is really perfect for that. And in fact, your digestive system will take starches and other nutrients, lipids, even proteins, and will break them down to try to get down to a very simple sugar that mitochondria can run off of. The secret of mitochondria is they want to run off about 3 carbon units. Glucose is perfect, it's 6 carbons. And I should define that for you now. So there's one here, so that's carbon 1, 2, 3, 4. Now there's one in the corner here, that's 5. And what's interesting, a lot of people will focus in on this and say, oh, carbon there. And I catch myself doing it every now and then. But not so. The last carbon, or carbon 6. Let's see if my stylus is going to be good for me today. My kingdom for a really good stylus. There we go. That's carbon 5. And that up there is the 6th carbon. So there you go. Important monosaccharide. We move on to fructose, and you might have heard of this as fruit sugar. You find it obviously in fruit quite a bit. Let's switch. This black is just a little too intense. There we go. This is a good, simple way of considering fructose. So as far as fructose goes, it's fruit sugar, so it's a very energetic molecule. So you're getting a lot of what the body needs to run your, I always think of ribosomes, but so fructose, being fruit sugar, is a very energetic molecule. Lots of carbon in there for our mitochondria to run off of. Fructose itself, you'll look at it at first and you'll say, hey, it's a pentose molecule. Well, it's pentose in shape, but if you count the carbons and go around, what you'll see is it literally has 6 carbons. It's a 6-carbon or hexose sugar. It just doesn't appear in its sort of appear in that way in its geometric shape. Now, galactose is an interesting one. I refer to this as glucose's cousin. It's like glucose, and you find this in a lot of milk products. In fact, galactose as a monosaccharide comes from consuming things like cheese and milk. It's one of 2 sugars that make up lactose, which is a 2-part sugar in milk. So when that 2-part sugar, lactose, is broken down, you tend to get things like galactose. Now, it's a lot like glucose. Specifically, what I'd say focus in on, as you go around the molecule in this direction like this, if you look at these groups, you say, well, okay, that's the same, and that's the same, and this looks the same, and we're same here. It's when you get over to this side, let's click accept there, where this is different, just flipped upside down. I'll accept that, and then we'll pop back a little bit to glucose. And if you look at it here, you'll see that arrangement. You can see the hydroxyl group facing down and the hydrogen facing up. And if you go to, let's go back, it's just the opposite here. So it's a heck of a lot like glucose, and our bodies can run off this. Glucose, of course, is very easy to take apart. Galactose, our bodies can digest it as well. So when they link up, you'll notice, I'll just highlight it again, right? Their left sides in cross section. There you go. Glucose and galactose. When these come together, and they can be stitched together by our enzymes, they make lactose. Now, when they come together, that's not a monosaccharide anymore. As monosaccharides come together, when you get two, you get a disaccharide. Monos, dis. You could say trisaccharide, but really, once you get more than two together, we just generally refer to them as polysaccharides or many sugars. So go through a couple of famous disaccharides that you would need to know. I sort of have some interesting little rhyming games here for these. A glucose makes a maltose, right? So when you put malt, for example, into sport drinks, things like that, it's a good source of glucose. It's just a disaccharide. And head to the mall, who doesn't like a malt, right? Now, if we come down and we combine glucose with fructose, well, you probably hadn't thought of that before. A glue-fru makes a sucrose. And that's one of the ways I remember. Glue-fru, su. Silly little naming games, but whatever works, right? So you need a hexose sugar, and you take another hexose sugar, because fructose is actually six-sided, and you get white granular table sugar. Excellent. Now, if you take a galac, you get a lac. And that's how that's worked out pretty well for students for a long time. Galac makes lac, and that's lactose, and that's what you'll find in high quantities in dairy products. And human breast milk is extremely high in lactose. Humans, unlike, for example, consider cows. Cows, when they're feeding milk to their babies, the requirement is that there's a lot more protein in cow milk than human milk. Cows have to get up, the little ones have to get up and move with the herd. That's all about survival. So when you look at the milk of bovines, it's extremely high in proteins like casein protein. And that isn't what human milk is all about. Human milk is more about supplying lactose sugar, and that's broken down very readily by our bodies. In fact, it's done by what we call the lactase enzyme. Neat thing about enzymes is they're often named after what they break down. So lactose, take off the ose, and the enzyme is called lactase. And that's what breaks it down. And when that enzyme breaks down lactose, well, you just get the monomers, right, or the monosaccharides produced. There we go. Should finish that off. So human babies should run in human formulas quite strongly concentrated with things like lactose. Giving infants cow milk, that's not the kind of milk that's intended for their development. Okay, it's not such a great idea, and there's a lot of protein there for a really juvenile digestive system to try to handle. So it's not really advisable. Stick to formula or breast milk. The added benefit of breast milk, by the way, is that let's just get this to full screen. I didn't realize we're in full screen. The added benefit of human breast milk is it's full of antibodies. It's basically like hooking up the baby's immune system to your own. As long as the baby is breastfeeding, they're getting the benefit of what your antibodies can do for them. It's sort of almost like being connected in a way, connecting the two immune systems, and it helps children to survive better. Just makes sense, right? Okay, so what do we have here? Let's see if I can shrink this down a little bit. There we go. How do these molecules come together? Well, coming together to build up is to synthesize. Oops. Hello. Let's have to go up here. Building up is synthesis. Now, it takes water. I sort of referred to that. It takes water to bring these molecules together or to take them apart. So let's consider the reaction arrow that is synthesis, and then I'll show you an animation of this. Now, synthesis, Let's go to a, I think I want to go to a, sort of a yellow. Let me get that where I want. Good. So here we are. Condensation synthesis. Now, condensation forming on your window, condensation, you think water. And synthesis is the buildup or creation with water. So, how does this work? Okay, well, here's the specifics. When you look fairly closely at the reactive ends, and that's exactly what they are, the reactive ends of our molecules, glucose, two glucose molecules, if you bring them together, you notice that the hydroxide groups are adjacent, right beside each other. And when you look at hydroxyl groups, you say, that's interesting. They're fairly reactive, and if you take the hydrogen from one group and the hydroxyl group from another, that would form water. If you just remove that, you get a water molecule. And that's exactly what's going to go on here. There is a link created by an enzyme. So basically an enzyme comes in here, reaches across to both, hard to dry hands with the stylus, but that's a little bit better, right? And brings them together. Enzymes are what are called catalysts. They make chemical reactions happen with a lot less energy. So in this case, we're going to condense off water or remove it, and its removal will bring the monosaccharides together. Let's pop out here. Now all that would be left, you see that sole, solitary, lonely little oxygen over there? Well, that's him over here. And that bond brings together the two monomers. And off to the side here, oops, undo there. Off to the side here, you'll see that water has been shed. There it is, right? One water molecule. And now these molecules have been linked. So glucose and glucose have been brought together. So we've synthesized a maltose disaccharide. Now, you can call this condensation synthesis because water is used to build up the molecule. It can also be thought of as dehydration synthesis. We think of dehydration as the loss of water. So there's another definition for this. So what I'm saying is they're synonyms. Dehydration synthesis, condensation, right? When water being removed to build things up. Same, okay? So don't get confused by that. I would accept either on an assessment. I learned dehydration synthesis. It doesn't matter. It's just another way of thinking about it. And there's our subsequent disaccharide right there. And this is called, this is an alpha linkage, just in case you're wondering. But you get more of that in biochemistry. We have enough terminology already, so I don't really want to get too terminology heavy with you. Maltose, the disaccharide. Yay. Moving on. Lactose is an interesting one. If you look at this bond, previously our bond between our monomers was like this. Okay? And that bond is easy for enzymes to get in and build up and to also disrupt or to remove, to cut. Now, lactose is a little bit of a different animal. If you take a glucose and a galactose and you bring them together, shedding water, of course, to make it. A glu-galac makes a lac. This, for example, this disaccharide is created in the breast tissue, right, in the mammary glands, and that is a devil of a link. It's a bit of a what we call a beta link, and it's hard for a lot of enzymes to get in there. Now, we do have an enzyme to get in there called lactase, and it does a pretty good job. As we age, we tend to produce a little bit less of this enzyme. And again, ACE for enzyme. Some people are lactose intolerant. They don't have enough enzyme to get in there, so they'll take things like lactate, etc., to supply enzymes so that they can consume dairy-based ice creams, obviously cream, milk, and even cheese. Because if you don't, if this isn't digested, it will ferment, and product of fermentation is a lot of carbon dioxide. There's a lot of bloating, a lot of gas creation. Some members of the population just naturally have a hard time with this. You see a lower prevalence of the lactase enzyme in African American, African Canadian populations, for example. It's an interesting molecule, lactose, obviously an important energy carrier and supplier, but it can be a little bit of a devil to get into. And this is neat because it builds up a little bit of what I want to talk about later when we get to discussing cellulose, because cellulose is another one which has bonds that, well, we just can't get in at all, right? And last time I checked, we can't digest grass. And that is classic cellulose. So the complex carbohydrates, when you go a little bit further and you start to put the monosaccharides together, you get long chains. And at that point, we're talking about polysaccharides. So I just want to pause, just remembering something here for you folks, that I wanted to show you on the course site, the synthesis here. So before we get too far into polysaccharides, let's see how those polysaccharides come together. Now, here's a nifty little animation here. I'm just going to mute that to show the condensation synthesis of two hexose sugars coming together. So the hydroxyl groups are very reactive, as I've said, and the formation of the bond at the exposed site, that's it right there. Now, if you think about this, if you pause for just 1 second, you realize that you could break these apart by putting the water back in there, reforming the hydroxide groups. So that comes up in a minute. But if you can remove water, you can add it too to cut this apart. So condensation synthesis is the removal to build up disaccharides and polysaccharides, things like that. But consider this. If you introduce it, well, that's something different. That's called hydrolysis, or cutting with the introduction of a water molecule. It's essentially putting the hydroxyl groups back in. But I digress. There we go. We'll consider the first case there. Now, here's the hydrolysis. Now, think of it as working in reverse. So when it comes to breaking apart, let's say lactose sugar, this is what lactase would do. There you go. And now you've got the monomers, or the monosaccharides. That's a neat little animation. That's on the course site. I suggest you view it a couple times and, pardon the pun, but digest this process. Because condensation synthesis builds and hydrolysis breaks apart. There's a neat little one here. There you go. You can sort of see the pairing together of the hydroxyl groups, and you go from a series of small molecules to big ones. Now, that went a little quick there, so we'll just watch both. Bring them together. There's a shedding of water. So what now you're making is a polysaccharide. Now, here's hydrolysis in the reverse. I should have paused it. So there's our little alpha links there, and if you hit play, the introduction of water by an enzyme to break it apart. Now, you have to break it apart because your mitochondria need smaller subunits. You can't run off these sort of long polysaccharides. They have to be broken down, which is why we have enzymes. Pretty neat stuff. Okay, so I'm going to keep this image here. It's important. It will come up again because we need to talk about what our polysaccharides are. Let's see here. Turn on mirroring, and we are back. Excellent. Alrighty. So complex carbohydrates, you hear that all the time. You hear that in diet-related sort of mediums, and you you don't want to have simple sugars, or you talk about eating white bread, and oh my goodness, that digests so easily that you flood your blood with too many glucose monomers. And it is true. Like if you consume things like white breads, they themselves they're just such they're long chains of glucose subunits, and they can be broken down pretty fast. However, if you eat things like pumpernickel, rye bread, etc., there's a little bit more in there to break down. It takes a little bit longer. It's a little bit more of a challenge for our digestive systems. So the supply of sugars is steady, but it takes time, right? You don't just get a sudden sugar rush as if you've eaten a Twinkie or something. However, complex carbohydrates, they supply us with energy, and we have to store them. We need this energy to be released all day long. Now, plant carbohydrates are, well, this is what plants do with photosynthesis. They build up long glucose chains, pardon me. So they stitch glucose together. And if you keep putting the glucose units together again and again and again, you start to get a long polysaccharide. And that's really a solar process. If you think back to it, that's photosynthesis. Let's just remind ourselves of what that is. And now I won't write it as a balanced equation, but take a dash of carbon dioxide, take some water. Now those aren't going to come together just by chance. It's going to take a little bit of energy to rearrange these bonds. So let's add some sunlight, and of course some minerals and things like that. But we'll just keep it simple. And you get a glucose molecule, right? Which just quickly draw like this as a hexose molecule, just for the sake of space. Now, if you can start putting those together, right, start making chains of those things, then what you've created is, let's see here, be good for me. It doesn't want to do it. There we go. You get stored energy. Just being a devil about giving me my last side. There we go. You put these together, right? And that's right there. I've just drawn more or less maltose. But if you keep going, if you keep making the chain longer, you're into a polysaccharide. Now, plants put together glucose in a beautifully ordered way. Animals, when we put glucose together, it's in kind of a chaotic sort of disarray. Well, I wouldn't call it not so much a disarray, it's just so highly branching. And we put lots of branches in because, well, animals move, and we need to have lots of these glucose molecules readily accessible. So we want it to be sort of a large sort of branching molecule, so there's lots to cut and there's lots of glucose for us to consume because we move very quickly. We just have greater energy demands than plants do. So pop out just a moment. There we go. Turn off mirroring. A little bit of a balancing act when I do this, but this is what I'm talking about here. When you look at starch, you'll get the long sort of subunits there, and then you'll get these bridges down here to the 6th carbon, and that'll continue. You'll see the chain. You can see how this would carry through, and they're long and row-like and orderly, and that's amazing. Now, when you look at plants compared to animals, we do a lot more branching. It's just a much more branched molecule. In fact, just pop out for 1 second here and show you what, let's see here. Glycogen looks like. Now, we'll just go to Google Images, and glycogen itself, this is a good little view of it here. There you go. You can see the highly branching nature of the molecule here. And that just supplies us with a lot of sort of immediate energy. I just wanted to show you that. If you look at, let's go back to Google for one moment. Okay. If you look at starch, not starch granules, You'll tend to find them in chloroplasts. Chloroplasts, are, after all, are where the sugars are made. And as you build up the sugars, you start to build up these starch granules. Now, when you look at a chloroplast, this is an electron micrograph of one. Let it come into focus. There we go. These little bodies here, these little oval-shaped bodies, this is starch storage. Here's another good one right here. So if we go back to our cell biology unit, there's the stacks of the grana, right? Those are the thylakoid membranes with the chlorophyll, and that's where, in the membrane itself, in the chlorophyll, that's where the photosynthesis reaction is happening. Now, the buildup of sugars will prompt the creation of glycogen. And here's some, here's a really nice rendering here. Right? You can see the membranes and the glycogen formation. And when the plant begins to this becomes quite prevalent, it will store it. And if you think about starch storage, just consider potatoes, for example. Right? Send those sugars. Looking out my basement window here, I'm looking at a couple of apple trees. Well, where's all that sugar storage going? Well, some is going out into the creation of those apples. So, not like it's not to be expected, but it's a smart thing. And it provides energy for the rest of us on the planet, because plants are the base of the food chain. So glycogen is really just animal starch. Now, you might wonder, Mr. Bleeker, what do I use for a title? Do I use polysaccharide or do I use starch? Well, I'm okay with you saying both. Animal starch, plant starch. It's all good. So there's a nice view of it, and you can see that these bonds, I'm just going to focus in on these alpha linkages down here, they're all very regular. And that's good. Occasionally, right, you get the links to adjacent rows of the molecule. Totally makes sense. Now, that's the kind of starch that we can consume. Not a problem. You eat potatoes, it's like that. You eat starch from rice, it's like that. You have pumpernickel, rye, whatever. You've got polysaccharide starch that works this way. After all, it came from plants. But then you look at this starch, which you can cleave very well, right? This is the animal starch. So there's lots of, let's see if we get the enzyme in there. There's a tremendous energy source here. Look at all those glucose subunits, right? There's one that we've left out, though, and it's going to come up in a minute where we talk about cellulose. Here's another neat shot of glycogen. And you can see in the muscles, there's the granules, these little dark spots, whereas it's much more sort of concentrated here in a much more orderly fashion, for example, than chloroplasts. So you can see how the molecules themselves are suited to different purposes. Now cellulose, let's draw this in. I'll choose a different color. I think a blue would be warranted here. Now you see how in this case the bond here between the subunits is facing up, whereas this bond is facing down. Well, this is what we call sort of an irregular pattern. Just a sec here. Pop back. These are all facing down, right? And these bonds alternate up and down. Now, what we've got here are a combination of alpha linkages and beta linkages. And that's some pretty insider biochemistry type stuff. What does it mean? It means that now you've got a problem, like this one, for example, will face down. You would have to have quite an enzyme to be capable of going in between these bonds and dealing with this problem. It makes for sort of a shorter molecule. It's a little bit tighter for space to get in there. And an enzyme has to be able to do a lot more. And when you look at it, like, well, our regular enzyme, for example, the one that's in your saliva, known as amylase, it can't get in here and do this job. It's not the right kind of starch digesting enzyme for this. In fact, the enzyme that would do the job for this, you'll find in the gut of a termite. Now, it's not the termite that actually makes this enzyme. The enzyme that digests cellulose, let's write ASE, is cellulase. Now, that's made by a little resident protist. You might remember the one called trichonympha from Biology 11. But the protist itself, the little one-celled critter, right? It makes the enzyme. The protist just lives in the termite gut. Now, there's sort of a mean trick that you can do with termites. If you sterilize their gut cavity and kill the protists, they'll digest cellulose materials. For example, termites are a real problem down in Los Angeles, a huge problem down in warmer climates because they're busy digesting people's homes. And if you irradiated their guts and killed that little protist, they would just fill up on cellulose and just die. They would be unable to digest it. Now, if you could pull this apart, you can get sugars, right? Think about this. If you could eat grass, you could get this specific series of monosaccharides out, but you can't because you don't have the right enzyme. So when we consume things like celery or roughage plant material that we can't consume, we just call it dietary fiber. It just goes through us, and that's healthy, right? Cleans out your system, kind of like swallowing a test tube brush, and keeps us healthy, right? Dietary fiber. Now, if we could break this down, we'd have another source of nutrients, but we can't. So there's a considerable amount of plant material that we cannot consume. There you go. Little, when we look at these linkages, kind of drawn in a little bit, kind of an interesting way here. When you look at the cellulose bonds, they're a little bit more angular if we look at it a little bit more up close. Starch bonds, they're wider. It's easier for the enzyme to get into.